Dr. Iraval blinked slowly as the genomic matrix flickered into resolution. He stared at the chromosomal alignment floating in the hollow dissection grid and frowned. Or the closest physical approximation his species could manage. There was something fundamentally wrong about what he was seeing. Strands of adenine and thymine, guanine and cytosine, twisted in the expected double helix, but they were not elegant. The human genome was riddled with what Iraval identified as errors, repeated sequences, inactive transposons, broken genes, redundant strands, and large non-coding regions that seemed to do nothing at all. Mutations scattered through the blueprint, some benign, others less so. Even within the so-called functional genes, there were misprints, base pair substitutions, frame shifts, insertions and deletions. It was a mess. The rival zoomed in on chromosome 17, trying to make sense of the BRCA1 gene. Sequences that would have been lethal in other species persisted here, sometimes passed down through generations without elimination. On his own world, such biological inefficiency was culled by engineered correction. Imperfection was a flaw, not an asset. "Doctor," said the assistant drone hovering beside him, "human subject has arrived." A rival turned from the sequence and stepped into the observation chamber. A man stood on the other side of the transparent barrier, arms crossed loosely, wearing the loose, standard-issue station garb of the Galactic Exchange. His name was Mason Clay. Mid-30s. Pilot. Volunteer genetic subject. Some injuries in his file, including mild radiation exposure and one punctured lung from debris during an asteroid interception. He moved with a casual limp and always had the expression of someone who'd seen enough to stop being impressed by most things. The rival activated the comm. "Mr. Clay," he began, voice clipped, "I have reviewed your genetic structure. It is flawed." Mason blinked. "Flawed how?" "You carry 4 hereditary mutations. Your Y chromosome exhibits partial microdeletion. You have 2 inactive viral fragments in your genome, 3 segments of broken mitochondrial inheritance from your maternal line, not to mention unexpressed duplications of defunct retroelement." Mason's mouth twitched. "Yeah, sounds about right. You say that casually, because that's how we're built." Iraval stepped closer to the interface. "Your DNA resembles a library that has been repeatedly flooded and patched with duct tape." "We like duct tape," Mason said. "It's flexible, cheap. You'd be surprised how much you can fix with it. But this," Iraval gestured to the screen, "is not fixed. It is unstable." "And yet here I am," Mason said, spreading his arms. "Standing, breathing, functioning. Why does your species not correct these errors?" "Because those errors are part of us," Mason replied. "They're not mistakes. They're chaos. And chaos is where the interesting stuff happens." Iraval tilted his head, confused. "Chaos impedes stability?" "It fuels change." The researcher pulled up the human genome overlay. "You carry a mutation that predisposes you to hypertrophic cardiomyopathy." "I passed my last flight physical," Mason said. "No signs of enlargement. It's a risk, sure, but so's walking out the door." "Mutation increases variability," Iraval pressed. "Too much and the system fails. Too little and the system never adapts," Mason shot back. "Your species edits everything, right? No disorders, no mutations, everything pristine." "That is correct. We are genetically optimized. Nothing is left to chance." "And that's why you stagnate." The room fell silent. Iraval stared, not out of insult but intrigue. Mason leaned against the wall. "See, humans are messy. We carry junk in our genes, useless code, broken loops. But every now and then that garbage throws out something new, an immunity, a tolerance, a better way to clot blood or resist radiation. It doesn't always work, but when it does, it sticks." Iraval blinked slowly, internal processors churning. "Your strength lies not in precision, but in redundancy?" "And in failure," Mason said. "Fail enough and eventually you find a new path." The medical tech interrupted then, signaling the next step in the evaluation. Mason was led to the diagnostic chair, where he extended his arm for the neural scan cuff. Aravale monitored silently as the cuff tightened, reading cellular turnover rates and hematopoietic stem cell activity. You regenerate damaged blood cells faster than projection, Aravale noted. There is a polymorphism in your JAK2 gene, but it appears to accelerate recovery. I got that after the asteroid hit. Radiation scrub changed something. Yet you improved. That's what I'm saying, Mason said. We break. We rebuild. Sometimes we rebuild weird. Arrival left the chamber in silence and returned to the analysis lab. He pulled up the genome profiles of 3 other humans on station, including one born with a spinal defect corrected through early surgery and another with a metabolic disorder managed with daily supplementation. Each genome was unique, messy, filled with contradiction. Yet they survived. He tapped a line of text into his research log. Human genome, not clean, not efficient, not optimized, but resilient through imperfection. Variation as a survival mechanism, mutation as a long-term strategy. And below that, without thinking, he typed the human's words. That's where the chaos lives. Aravale did not sleep in the traditional sense, but he spent the next 4 rest cycles deep within the genomic archives, comparing species profiles across 400 known oxygen-breathing organisms. Most advanced civilizations had long since pruned their genetic code, either naturally or through generational intervention. Their sequences were symmetrical, predictable. Redundancies were removed, instabilities were erased. Then there were humans. They didn't just tolerate errors, they accumulated them. He opened a new case file. This one belonged to Naomi Kessler, another human volunteer, civilian background, age 49. She carried 2 known single nucleotide polymorphisms that increased her risk for certain cancers, a 32 base pair deletion in her CCR5 gene and a minor translocation on chromosome 9. She also had never been seriously ill in her life. Her T-cell response was double the station median. When challenged with a synthetic viral antigen during standard immunoreactivity trials, her immune system mounted a defense so fast it had to be slowed pharmacologically to avoid outpacing the control algorithm. Arrival summoned a 3D representation of her immune profile and stared in disbelief. There, embedded within all the broken code and unnecessary repeats, was a system that overperformed expectations. A deleted receptor gene, one that should have impaired her, actually enhanced her resistance to several viral loads. Damage, he whispered aloud, becomes defense. He summoned Mason Clay back for a follow-up. The human arrived 20 minutes late, limping slightly from a joint strain sustained in a climbing drill. He looked tired. Aravale noticed subtle inflammation markers in his gait and mild adrenal elevation. Yet his eyes were clear, reactive. Your body is undergoing multiple minor stress events, Aravale said. You are compensating. Yeah, I'm always compensating, Mason said, dropping into the chair. That's life. Aravale tapped the console. Explain something to me. You accept internal errors as routine. You build systems atop unstable foundations. Does this not alarm you? Sure it does, Mason said. But the thing about chaos is, once you learn how to navigate it, you stop expecting it to be neat. You start treating it like a partner instead of an enemy. I do not understand. Mason leaned forward. You see those mutations and broken sequences as disorder. I see them as potential. Most of it doesn't lead to anything, sure. But sometimes a kid's born with an adaptation no one saw coming. Extra pain resistance, better muscle recovery, cognitive advantages, creativity we can't explain. We don't get that by editing things to perfection. We get that by leaving room for surprise. Surprise is dangerous, Aravale said. It is how civilizations fall. Or change, Mason countered. Nothing grows without risk. Aravale studied him. Your view of biology is philosophical. It's practical, Mason said. You think we should fix every flaw in our genes, but the flaws are us. That unpredictability is what lets us adapt to things no one planned for. That's why you bring us along on colonization runs, right? Because when the unexpected hits, you need someone who doesn't panic, someone whose cells already learned how to adapt the hard way. Aravel paused. You believe your species is chosen for resilience? Mason nodded once. Because we don't have the luxury of being tidy. Derival returned to the lab. Over the next 2 days, he reviewed records of human genetic disorders: Duchenne muscular dystrophy, cystic fibrosis, sickle cell disease. But for each, there was a counterbalance. New treatments emerged. Carriers often showed resistance to unrelated infections. Traits once seen as failures became shields, adaptations built on top of mistakes. And then he found the case that crystallized it. Patient 63,821, male, 22 years old, radiation accident survivor. Severe DNA fragmentation. Expected outcome, terminal failure of replication machinery. Instead, his cells developed novel repair mechanisms, unstable but efficient. He lived. 6 years later, his offspring inherited a version of that system, weaker but still functional. An accident created a mutation. The mutation created a new biological tool, and the system adapted. The Ravel wrote for hours, transcribing data, cross-referencing every failed repair and every unlikely survival. Patterns emerged, not of control, but of possibility. The human genome wasn't just a blueprint. It was a living record of experimentation, rewritten by error, sustained by endurance. His final note that cycle was simple. Human DNA is not clean because life isn't clean. The mess is the point. Erivol's lab had changed. There were new sequences mapped along the walls now, drawn not in order but in sprawling constellations of influence. Mutations here, adaptations there, with threads connecting unrelated genes across generations. The chaos was no longer an obstacle. It was a map, an unruly, branching mosaic of the human genome's defiant resilience. Mason returned, this time for a comparative scan. Aravel wanted to track somatic mutations over a 5-year span using stored medical data from Earth. Standard longevity research. Mason just wanted to get back to flight readiness. Blood works clean, Mason said, watching the screen while Ervile moved the scan node up his arm. Heart's fine, liver enzymes are in range, so why am I grounded? You exhibit elevated protein markers related to inflammation, Aravel said. Likely microtrauma from soft tissue strain. Not dangerous, but it indicates recovery lag. Mason groaned. My body's always lagging. That's normal. Iriable tilted the scanner slightly. Your normal includes 5 overlapping gene activations that are usually suppressed. You're running at levels most species would consider pathological. And I can still bench my own weight. That's the thing. Our genome allows us to walk a tightrope. Not everyone balances successfully. No, Mason admitted. But that's part of the system too, the fact that we don't all handle things the same way. You want consistency. We survive by exception. Arrival recorded another note. Your exceptions lead to damage and discovery. Ask any geneticist back home. Some of our most important insights came from people with conditions no one understood. Mason lifted his sleeve and tapped a faint scar on his forearm. I got an experimental treatment for tissue regeneration. It was risky. They used engineered stem cells cultured from someone with a collagen disorder. It shouldn't have worked. But the rest of the galaxy believed. Better than anyone expected. My muscle repair time dropped by 1/3. Aravel saved the scan data. He stared at the graph displaying Mason's recent cell turnover. The line was jagged, unstable, but functioning. After Mason left, Aravel ran a projection model. He simulated what would happen to a human genome if all the errors were corrected, cleaned, organized. The result was terrifying. Uniformity, precision, and total vulnerability. Any unexpected pathogen, environmental shift, or injury would cascade without redundancy or fallback. The genome couldn't bend anymore. It would break. He ran another model. Left the junk code, the repeats, the mistakes. The cell response time dipped slightly, but the overall survival curve extended under stress. Potty code died faster. The following week, Naomi Kessler returned for a muscle biopsy. She had agreed to assist in a study of mitochondrial variants in tissue-specific gene expression. The probe collected samples painlessly, and Erival began sequencing within minutes. He was looking for an answer to a different question now. Not, why is this here, but what else could it do? In Naomi's sample, he found a near-silent strand, a fragment of non-coding RNA buried inside a sequence of broken viral DNA. At first glance, completely irrelevant, but when simulated in stress environments, oxidative overload, thermal shock, the fragment activated a tiny, previously unrecorded protein cascade, protective, not permanent, but potent. A gene remnant from a virus long thought extinct had saved the cell from catastrophic stress. She didn't even know she had it. When Erival told her, she laughed. Figures. My grandmother survived a gas leak in a copper mine, came out coughing but never got sick. Maybe it started there. Erival began reconstructing her maternal line's genetic record. 2 generations of miners, 1 radiation-exposed nurse, 1 pregnancy during famine. Her family history was written in survival events. The mutations weren't random, they were earned. Her blood didn't just carry errors, it carried a history of solutions. That night, Erival stood before the board of xenogenetic integration. 10 delegates, 5 species, all with optimized code, stable inheritance systems, and centuries of eugenic precision. He showed them the chaos. He showed them the deletions, the repeats, the duplications. And then he showed them what that chaos did. Increased resilience, fast adaptation, uncommon resistance. Human DNA, he concluded, is not a stable code. It is a volatile language, not written to be understood, but to be reinterpreted. The delegates questioned, pushed, demanded projections, asked why such chaos hadn't destroyed the species. It nearly has, Erival answered, but each time it rewrote itself faster than the crisis could finish. He paused. They don't evolve because it's time. They evolve because they have no choice. The board allowed silence, then tentative approval. Erival returned to his lab, updated the species classification tags. Human, genetically unstable, environmentally adaptive. Primary survival feature, biochemical improvisation. At the bottom, he added Mason's words. It's where the chaos lives. The damage to the outposts began with a microfracture. A minor seismic tremor below the terraforming station cracked one of the geothermal regulators and sent a cascade of mechanical faults through the subterranean support lattice. At first, it was background noise, environmental compensation. Then pressure rose, heat spiked, and the automated suppression system failed. When the emergency alarm sounded, there were 6 humans on the station, including Mason Clay and Naomi Kessler. They were the ones who ran toward the sublevel breach, while others scrambled for evacuation. Erival watched the footage days later. The humans didn't hesitate. Mason moved through smoke and rising temperature with his shoulder braced against a misaligned corridor door. Naomi, coughing from irritant gas, still managed to extract the coolant lines manually after the control servos had locked. When they emerged, Mason's skin was blistered along one arm. Naomi's oxygen saturation was dangerously low. Neither of them asked for help until the station was stabilized. It took 31 hours of regeneration therapy before Mason could fully extend his left arm again. The upper layers of his dermis had sloughed off, and the underlying tissue showed signs of thermal protein degradation. Erival visited his recovery chamber on the 2nd day. Mason sat propped up in the medbed, one arm in a biogel sleeve, heart rate steady. A faint IV line traced up from his wrist to a compound mix of electrolyte rebalancers and metabolic accelerants. His body was healing, faster than baseline. The gene activations were subtle but unmistakable. You should not have withstood that much exposure without long-term tissue loss, Araval said, reviewing the logs. Wasn't thinking about my cells at the time, Mason replied, his voice dry but steady. Just didn't want the core to rupture. Araval set the data tablet aside. I analyzed your repair patterns. Your fibroblast activity is 10% above human medium. Your inflammatory suppression genes upregulated within 2 hours. This shouldn't have happened. Mason raised an eyebrow, but it did. I believe you possess a rare mutation in your HSP70 pathway. It may have activated post-trauma. Mason nodded. So the burn woke something up. It's not just you, Araval said. Naomi's lungs filtered 3 times the volume of particulate toxins than our models predicted. Her alveolar membranes showed pre-existing reinforcement, not from genetic engineering, but from a protein expression sequence likely inherited. She'd never seen those toxins before. She didn't need to, Mason said. Someone in her bloodline probably did. Her rival paused. She adapts because her family already survived something similar, he added. Mason smiled faintly. That's what I've been trying to tell you. We carry more than just our past. We carry all the tries that didn't work. And sometimes, one of those old failures lines up with a new problem. Araval stared at him, then walked to the screen and pulled up Naomi's chart. The same sequence. A once dormant section of code inherited 3 generations back had activated in response to the synthetic hydrofluorocarbons. An environmental crisis had awakened a solution, buried in a line of unused code. It wasn't design. It was memory. A week later, the station was restored. Araval presented the findings at the Galactic Biogenetics Symposium. This time, he didn't bother with disclaimers. Human DNA is a record of mistakes, he said, but those mistakes are not waste. They are contingency, redundancy, adaptability. He showed time-lapse recovery footage of Mason's arm, overlaid with gene activation points. He played alveolar scans of Naomi's lung response, matching each phase to archived trauma logs in her lineage. And then he said the thing no one had expected him to say. We should not edit them. The hall fell silent. These errors, they are not the opposite of order. They are a form of biological wisdom. We do not understand them, but they do. He ended with a final annotation on the central genetic display. In humans, chaos is not failure. It is a strategy. Afterward, Araval returned to the observation deck. Mason stood there, healed arm folded across his chest, watching the orbiting planet rotate in slow amber arcs. Did they buy it? He asked without turning. I do not know if they understood, Araval said, but they listened. Mason smirked. That's a start. Araval joined him at the rail. Your genome is a battlefield. It is full of scars, yet you survive. Because we don't erase the scars, Mason said. We learn from them. Araval nodded. Later, in the solitude of his quarters, he recorded a personal note. Humans thrive not despite their biological flaws, but because of them. Their imperfection is a form of intelligence, a defiance against finality, a refusal to settle. And somewhere in their disorder lies a kind of genius that cannot be replicated. He closed the file with a single phrase. They do not carry chaos, they are chaos, and somehow they make it work.